Assalamu alaikum. This is Dr. Saniya Shuja uh, from Shalamar Institute of Health Sciences. Um, Today, I'm going to talk about thyroid papillary carcinoma with BRAF mutations and their association, with the iodine resistance and some studies related to it. The topic of my today's discussion is from molecular changes to therapeutic impacts induced by BRAF mutation in papillary thyroid carcinoma. There are no conflicts of interest to report. And at the end of these presentations, the audience will be able to reflect upon iodine trapping, transport, factors influencing these processes, etc. Understand and apply the knowledge regarding downstream effects of BRAF mutation on MAP kinase pathway. Apply knowledge regarding in vitro and in vivo effects of BRAF mutation to clinical disease. Discuss patient management issues in relation to iodine resistance in differentiated thyroid carcinoma and papillary carcinoma, and the rationale for using inhibitors, namely BRAF inhibitors or MEK inhibitors in thyroid papillary carcinoma to enhance treatment with radioactive iodine. So in order to understand the subsequent subject, I would just like to throw some light on thyroid and iodine. Thyroid contains 70 to 90% of iodide in the body, and this iodide accumulation is dependent upon sodium iodine symporter, which is expressed on the basolateral membranes. These are the portion of the membranes that face the interstitium of thyroid follicular cells. And the iodine in the blood is taken up through sodium iodide symporter into the follicular cells, where its level is about 30 times that of in the blood. And this 30-fold increase in iodide concentration in the thyroid follicular cells is done by sodium iodide symporter, which is dependent upon the electrochemical gradient of potassium sodium ATPase. And this NIS symporter, it takes in 2 sodium and 1 iodide molecules. And this iodide molecule, when taken up into the follicular cells, it is then released to the lumen across the apical membrane via pendrine, which is an anion transporter. And this process is regulated very closely by thyroid stimulating hormone, and which regulates sodium iodide symporter, and also influences its translocation into the cell membrane, where it can be effective over there. Now, once the iodide reaches the cell colloid interface, it is oxidized by thyroid peroxidase, and it is with the help of dual oxide type 2, and then it is incorporated into tyrosine residues to form a monoiodinated tyrosine residues and diiodinated tyrosine residues. And this process is called organification and coupling, and this coupling is done by thyroid peroxidase. So in this figure, you can see that this is the iodide, and then its incorporation into the MIT and DIT, which are the mono and diiodinated thyroid oxidase. So this schematic representation gives the overall view of this whole process. Blood in the iodine is taken up by the follicular cells, 30-fold increase in concentration through the sodium iodide symporter. And this is dependent upon the sodium potassium ATPase pump. And once here, the iodide is then, it goes into crosses the apical membrane to go into the membrane colloid interface where it is oxidized, and then becomes a part of colloid, which is thyroglobulin. And then, of course, thyroglobulin, under the effect of TSH, once again, is pinched off and taken up by thyroid follicular cells, and then T3 and T4 are released in the blood. So now it is that because of this high concentration of iodine, you know, thyroid PTCs or papillary thyroid carcinomas and differentiated thyroid carcinomas, they also have higher concentration of iodide, and this serves as the basis of treatment of metastatic disease by iodine 131. You know, of course, you ablate the thyroid and then you give iodine 131, and the metastasis, they concentrate this radioactivity, and then the cells are killed, and of course treatment takes place. But it has been brought to the notice of the clinicians that many DTCs, differentiated thyroid carcinomas, are incapable of concentrating iodine to a level to be sensitive to treatment with iodine 131. And so the question arises, why and how do these tumors get resistant to iodine, and then therapy becomes ineffective or partially effective? Among them are a percentage of conventional and follicular variant of papillary thyroid carcinomas. Also, tall cell variant is also partially resistant to iodine. And 61% of these have BRAF mutation, even though these tumors also have a TERT mutation. Then is hobnail or micropapillary variant of PTC, which has 80% of them have BRAF mutation. Now, several studies indicated that BRAF mutation played a role in tumor growth and prognosis and resistance to iodine therapy in both PTC and DTC. So I'm going to go over some interesting studies. The first one is by Zing et al., who studied a total of about 1,800 patients from 13 medical centers in 7 countries. And they found that BRAF mutation was seen in 45.7% of these papillary thyroid cancers. And interestingly, they also noted that those patients with BRAF mutation had higher stages of cancer. And this suggested that BRAF was somehow giving a more aggressive growth pattern to the tumor. And they concluded that these tumors which had BRAF mutation, they probably required more aggressive therapy. The other study by Zin, Liu et al. Is a meta-analysis in which they searched Medline, PubMed, and Web of Science, and pulled out a total of 69 studies involving 14,000 patients. And their results showed that BRAF mutation was associated with extrathyroidal extension, advanced TNM stage, lymph node metastasis, multifocality, and recurrence. And thus, their conclusion was that BRAF mutation, it provides prognostic information also for papillary thyroid carcinomas. This is a Kaplan-Meier curve, and it gives estimate of recurrence-free probability of papillary thyroid carcinoma in patients with and without BRAF mutation. So as you can see here, on the y-axis is the recurrence-free probability, on the x-axis is the months, time period in months. And you can see that the BRAF negative patients are faring much better, almost like 70% recurrence-free probability at about 108 months, compared to BRAF positive patients who had almost like 40% or less than 40% disease-free probability at about 108 months also. The second Kaplan-Meier graph gives a comparison in Korean series, in which again they found that at about 120 months, the BRAF negative patients had almost like 90% recurrence-free probability, while the BRAF positive patients had less than 80%. Thus, this also suggested that BRAF somehow gives more, uh, is gives a worse prognosis. Now, the natural question arises, why is BRAF mutation associated with increased aggressiveness and recurrence of PTC when compared to red PTC, which is the second most common genetic alteration in PTC? And both of these are expressed through the MAP kinase pathway. So let's look a little bit more. This is a diagrammatic representation of the mitogen activated protein MAP kinase pathway. You can see here that upon activation and binding at the membrane, RAS protein interacts with BRAF on activation. And this BRAF again phosphorylates and activates MEK1 and 2, which in turn phosphorylate ERK1 and 2. And the phosphorylation of which leads to other downstream proteins which get phosphorylated, including many kinases, which overall leads to alterations in expression of genes in the nucleus, which are involved with cell proliferation, growth, survival, and tumor regenesis. Now, in comparison, if you look at ret PTC, what you see here is that it is having an effect on this MAP kinase pathway upstream of BRAF. And somehow this upstream effect is much weaker than the effect of BRAF on the MAP kinase pathway. Now, there were also some animal studies, for example, in rat thyroid cell lines. It was noted that BRAF mutation caused greater genetic instability in the cells. It also led to markedly increased matrigel invasion as compared to red PC. And of course, so in an animal model also, it was concluded that BRAF mutation is probably a stronger activator of MAP kinase than red PTC. In human PTC tumors, there were studies about gene expression profiles, and they showed that those tumors which were BRAF positive had a very different genetic gene expression profile, almost like a gene signature as compared to BRAF negative papillary thyroid carcinomas. Now, I'll just refer to the study by a group, Oler group, in which they studied gene expression in papillary thyroid carcinoma, and they found some new transcripts. And these transcripts could then act as targeted molecules because of their overexpression in papillary thyroid carcinomas. So they looked through gene expression profiles in SAGE libraries from thyroid tumors in normal thyroid tissue, and also did quantitative PCR. And they found that there were 3 types of genes that were overexpressed, including CXCL14. And so let's look at further study of CXCL14. They found that if you look at the last 3 columns, that you can see here CXCL14 is expressed in this cell line as compared to the wild type where BRAF mutation has not taken place. You can see absence of this gene. By quantitative PCR, they found that CXCL14 was increased 30 times in papillary thyroid carcinoma when the BRAF mutation was homozygous as compared to the wild type when it was negligible. Thus, they went ahead and wanted to see what type of cells in the tumor are expressing this protein. And they did immunohistochemistry and found that these were the epithelial tumor cells that had the increased staining. You can see here brown, strong staining in papillary thyroid carcinoma. The stromal cells and other cells were negative. And so, of course, this can be used as a targeted molecule in future. So from all these studies, it is convincing that molecular events that are coupled to BRAF V600 may be responsible for tumor aggressiveness, which is uniquely associated with BRAF mutation. And also it can lead to expression of new types of genes. Thus, coming back to the MAP kinase pathway, we can say that BRAF may exert relatively strong oncogenic effect and thus pronounced molecular events coupled to thyroid activation, proliferation of neoplastic cells, etc. And also noted was that BRAF mutation was far more profoundly associated with decrease in thyroid peroxidase expression in primary PTC, as compared to when other mutations happen like RET PTC or RAS mutation in thyroid carcinoma. So BRAF can participate in thyroid tumorigenesis through a lot of ways, including tumor angiogenesis, upregulation of VEGF, aberrant gene expression, methylation, etc. And so coming back to iodine resistance in differentiated thyroid carcinomas and papillary carcinomas, we know that iodine resistance to iodine 131 makes it difficult to treat thyroid carcinomas. And some PTC are relatively more resistant from the outset, that is de novo resistance, while others develop variable resistance during treatment. And thus, we are in search for any treatment that can reduce the resistance of PTC to iodine 131 therapy. This could be MEK inhibitors, because as we saw, they are in the MAP kinase pathway. An interesting study by Rothenberg et al. Needs some mention here. And what they did was they used a BRAF inhibitor, dabrafenib, in patients who had developed resistance to iodine therapy, and found out that after giving this inhibitor for a while, the patient's tumors, they started concentrating radioactive iodine, and that led to dramatic tumor response. And thus, this leads to our concluding slide that BRAF mutation has profound effects on tumor growth, on radioactive iodine resistance, and using BRAF inhibitors can be helpful in making the patient's tumors again sensitive to iodine 131 therapy. Also, when we look at treatment and management issues, an important thing is to know the morphological features of PTC and differentiated thyroid carcinomas. For example, whether it's a hobnail cell variant where 80% tumors have BRAF mutation, so they are more resistant to iodine therapy. Also tall cell variant. And these studies also help us to find new targets for therapy. And this can be done in the form of gene profiling, etc. So hopefully we are on the right path for treating our patients with papillary thyroid carcinoma, and hopefully we'll find new markers on the way. Thank you.